All right, Ted, take it away.
Well, good afternoon, everybody. Our distinguished guest and our invited speaker this afternoon is Dr. Chelsea Wood. Dr. Wood is a professor at the University of Washington in the School of Aquatic and Fishery Sciences. Dr. Wood's research interests are centered on the ecology of parasites, pathogens, and the ecology of infectious diseases. She focuses in her research on both theory and practical application of ecology. And a lot of her work over the years has focused on the key general question of whether the incidence of disease, both in natural systems and as afflicting human beings, is linked in fundamental ways to biodiversity. And in particular, whether the loss of biodiversity generally results in increases or decreases in parasites and pathogens and the incidence of disease. So in yesterday's seminar, Dr. Wood focused on her research on parasites of marine fish, including her team's use of museum collections now to assess historical levels of parasitism over long periods of time, and her work on coral reef fishes and their parasites to assess how fishing affects host-parasite relations. We learned from Dr. Wood's research that a key element in the effect of fishing is the importance of the transmission strategy of the parasite. In today's seminar, Dr. Wood will focus on some of her work on parasites in freshwater ecosystems and also on her interest in seeking ecological solutions to disease problems. And in particular, she'll talk with us today about her ongoing work in Africa with schistosomiasis. So I'll turn things over now to Dr. Wood.
Thank you so much, Ted, for that lovely introduction. And thank you to all of you for having me here again. When it comes to scientific research, I've had two great loves in my life. The first you got to hear about a bunch yesterday. I'm deeply interested in, as Ted said, understanding what past ecosystems look like from a parasite's perspective. And that interest comes from my utter fascination with parasites, their diversity of body forms and life histories, and their apparent vulnerability to environmental change. But on the other side of this coin are parasites that just won't go away no matter what we do to them. And because while parasites are staggeringly beautiful, when you consider the improbable adaptations that they developed to survive, the flip side of that coin comes when these staggeringly beautiful parasites affect human hosts. And while I respect all parasites and love each and every one of them for the ways that they make this world weirder and wilder, there are some parasites that I think must be utterly annihilated, wiped off the face of the planet. And now I'll tell you about my team's small contribution to driving one parasite species into oblivion. Now, this talk is going to be comparatively short, both because it's dense. I'm going to pack a lot of data into 35 minutes, and because I want to leave lots of time for discussion. This is an active and growing part of my lab's research, and I find it really helpful to get feedback and ideas. So I hope you'll join in the discussion in the last couple minutes of our time here today. So let me begin by telling you that everything that we're going to discuss in this talk is going to come from my team's recent paper published in PNAS in December of 2019. And the work absolutely would not have happened without our very large team. I want to especially point up Sana Sokolow, Julio DeLeo, Izzy Jones, and Andy Chamberlain, who are at Stanford. And they were involved intimately in the conception and execution of this work. I also want to point to our on-the-ground colleagues at the public health nonprofit Espoir pour la Santé, our stats team of Grant Adams at UW and Skylar Hopkins at North Carolina State, and many others. Our work was supported with an NSF-coupled Natural Human Systems grant, a grant from the Bill and Melinda Gates Foundation, and support from the Michigan Society of Fellows at the University of Michigan. So it can be easy for us to forget here in the developed world about infectious disease. We no longer have to worry about malaria or cholera or sleeping sickness, but that doesn't mean that infectious disease has been conquered. It's still an everyday part of life for most of the inhabitants of this planet. And most of the infectious diseases that plague people have an environmental component to their transmission. And that means that ecologists could be the key to improving health outcomes. Now, physicians, doctors, have been rightly focused on individual human patients, but ecologists can help by broadening the view to look at the patient in the context of their local ecosystem. My team calls this strategy ecological levers for health, but the idea isn't anything new. To tell you about it, I want to take you back to Victorian London, to the mid 1850s. At that time, London was a teeming city and one that was racked by recurrent epidemics of this disease. Today, we know that cholera is caused by a bacterium, Vibrio cholerae, and that bacterium is ingested from drinking water that's contaminated by the feces of an infected person. But back in the 1850s, no one knew what cholera was or why it happened. Enter John Snow. Today, he is considered the father of epidemiology. But in 1854, he was just a physician living in London's Soho neighborhood, which in that year was racked by a massive cholera epidemic. Snow's innovation was to map the cholera cases in his neighborhood, and as soon as he built his map, it pointed him straight at the cause of the outbreak. It was just one pump at the corner of Broad Street and Lexington. And as the story goes, when Snow took the handle off that pump, the cholera epidemic stopped. Eventually, he figured out that the reservoir for the Broad Street pump was being contaminated by waste from a nearby building. So from its very birth, the study of disease has depended on explicit consideration of space. It's depended on maps. Since then, a lot has changed for London. But in other parts of the world, very similar problems persist. I work on a disease called schistosomiasis. We call it schisto for short. This is a worm that infects people, and there are two species that cause two different sets of symptoms. Urogenital schistosomiasis is caused by Schistosoma hematobium, and intestinal schistosomiasis is caused by Schistosoma mannsoni. This is the global distribution of cases of schistosomiasis across the world. Worldwide, there are about 206 million cases of schistosomiasis today, and that makes this the world's second most prevalent parasitic disease after malaria. Now, most people here in the US have heard of malaria because rich people occasionally get it. But few people have heard of schisto, in part because it affects the poorest of the poor, and the vast majority of those cases are in sub-Saharan Africa. We work in the West African nation of Senegal, where we do not need any fancy tests to tell us about the gravity of this disease. These are a couple of school kids who are enrolled in our study, and these are some representative urine samples that they might give us so that we can assess whether they're infected with urogenital schistosomiasis. Clearly, something's not right here. This is what's going wrong in the bodies of those infected individuals. Adult schistosoma worms take up residence in the veins around the bladder and the intestine, and there can be many, many worms in one human. Here you see a blood vessel in red. The shiny stuff is connective tissue, and then coiled up inside of those blood vessels are a number of adult schistosome worms. This is some video from the parasite ecology course that I teach every autumn. In one of our labs, the students get to dissect a recently sacrificed schistosome-infested mouse. And we kill those mice right before the lab starts so that the worms are still alive when the students do their dissections. And this is what they get to see: real live worms in situ wriggling around inside their host's blood vessels. And this is exactly the way that it would look in a human organ. Now, these worms have a big problem. They're producing eggs, which somehow need to make it into the outside world. But if everything is going well in your life, your circulatory system is not open to the outside world. And these worms have an ingenious and even a diabolical solution to that dilemma, which is that their eggs have these small spines. Those spines cause the eggs to become snagged in the wall of the blood vessel. They stimulate an immune response, and it's that immune response that helps transit that egg over the wall of the blood vessel and then through the wall of the bladder or through the wall of the intestine, where they can then be voided out into the world in urine and feces. Success for the parasite. But not every egg makes it successfully into the outside world. About 2/3 of them get stuck along the way in the wall of the bladder or the intestine. This is a cross section of the wall of a human bladder, and all those little football shapes are eggs. Now, because they're foreign objects, the body tries to wall them off. The body builds this calcified granuloma around those eggs. And if you get enough granulomas, they basically replace all the useful tissue, and that organ can't perform its normal function anymore. This is an X-ray of a urogenital schistosomiasis patient, and X-rays are supposed to show calcified structures. For a normal patient, that void in the pelvis should be totally black. Instead, we see that shadowy structure. That is the urinary bladder, a soft tissue organ that is now so calcified that it actually shows up in X-rays. And you can even see kind of the ghostly outline of the two ureters leading down to the bladder. That degree of calcification means that the bladder tissue is not able to do its job properly, and it's what causes that symptom of bloody urine. But those eggs can go anywhere. They're not limited to just the bladder, and they do damage to a whole variety of organs. And lest you think, oh, well, what's a couple of tiny worm eggs in your organs? This is what a patient with severe schistosomiasis looks like. That abdominal distension occurs because fluid builds up in the belly. It's painful, debilitating, and sometimes fatal. And the burden falls disproportionately on kids. This is a plot showing the prevalence of infection, the proportion of people infected as the red line, and then the number of eggs produced for every 10 mils of urine is shown in the purple columns. And the peak represents kids who are between 10 and 14 years old. That peak will shift younger and younger the hotter the transmission hotspot. It's a shameful state of affairs that this is happening in 2021. But how are these kids getting infected? Well, once those eggs are eliminated from the body in urine or feces, they hatch in freshwater and they infect freshwater snails. The snails are an obligate part of the schisto life cycle, and they are teeny tiny. Within those tiny host snails, the worms replicate asexually, and they send hundreds of thousands of clones of themselves out into the water column. Those clones are called cercaria. They swim around until they find a human to infect. You'll see some video of these cercariae soon. They're really cool life stages, just barely visible to the naked eye, and with this evil-looking forked tail. They penetrate the human skin, and then they take up residence in the circulatory system. Now, we've got a really good drug to treat schistosomiasis. It's called praziquantel. It's a cheap one-time dose that kills all the adult worms inside a person and totally clears them of their parasites. And you're watching the most common method of drug distribution. This is where a public health worker comes to a school and helps the school teacher dose up all of her kids. Merck CGA actually donates these pills for free. So why are over 200 million people still infected? The answer is that a cured person immediately becomes reinfected when they contact a contaminated water source. We've been dumping millions of tabs of praziquantel on Africa since the late 1970s, and in most places, the prevalence of schisto has not budged. The environmental component of the life cycle is what we need to address if we're going to make any progress here. And the place to start is by doing simple mapping, just like Jon Snow's maps of cholera. We need to better understand that spatial ecology of transmission. This is a typical water access site in Senegal, and you can tell from this picture that there's intensive water contact and multiple uses for the site. Now, say that we want to try and interrupt transmission at this particular site. We obviously need to be targeting the snails that are the source of infection for people in the environment. But where do we do that? Are the snails that produce parasite infective stages here at the site? Are they a meter away from these swimmers, or 10 meters, or a kilometer upriver? We don't know. Are they associated with a particular habitat at the site? Maybe mud or vegetation or the edge between the two? We don't know. Is risk evenly distributed across the site, or is it patchy? Are those patches associated with particular environmental features like shallow water, or maybe the infective stages of the parasite are emanating out from this emergent vegetation? We don't know. Schisto is an incredibly prevalent disease, and yet we don't know the answers to any of these questions. And without those answers, we have no idea how to break the chain of transmission. We don't know where the Broad Street pump is, and we can't remove its handle. Now, this is increasingly being recognized by the global health community. In 2016, the World Health Organization recognized the failure of praziquantel, and they started recommending a combined strategy, both give people drugs and control the snails. But where do we start with those snails? That's the question that my colleagues and I wanted to answer. How do we do smart snail control? And we have to do this in a smart way for a number of reasons. The first is that most snail control is accomplished with the use of chemical molluscicides. Niclosamide is the most common one, and it's not great to use in places where people are intimately interacting with water, especially where they depend on healthy and vital fisheries, like the villages where we work in Senegal. Now, snail control can be done chemically, or it can be done mechanically. You can actually remove snail habitat, but all that is expensive. And if there's one thing that all schistoendemic regions have in common, it's that they're all very poor. Now, of course, we wouldn't necessarily expect these very poor people to pay entirely for these interventions, but when you're asking philanthropists or funding agencies to pay for it, they want to see endpoints. And in most cases, dealing with schisto is like shoveling poop against the tide. So for all these reasons, we need to find ways to do snail control more efficiently. But we really have no idea where to start. And there's one more thing that you need to understand about schisto before I can tell you where our team comes in. And that's that in the past, the ecology of schisto has been studied in a really haphazard way. The vast majority of past studies have used timed searches to count snails in the environment. This is one of our technicians, Pabgai, demonstrating this technique at one of our sites. The traditional way is to attach a flower sifter to a pole and then go out and search for snails for 30 minutes. And obviously, this is not the ideal way to measure snails. It doesn't give you an absolute density. It's really dependent on the skill of the technician. It doesn't allow you to compare to sites outside of your own study, and it doesn't even really allow you to extrapolate from one part of your own site to another. And the result is that there are very few studies that have even found a correlation between the presence of snails at a site and the burden of human infection at that site. And in fact, tons and tons of studies report a total lack of correlation here. Is there no correlation because people aren't getting infected locally, or because the sampling strategy sucks? Here, what we have is a total blank slate. The WHO wants to do snail control, recognizes that drugs aren't enough, and yet we have virtually no information to start from. Where are the snails and how do we target them? Our goal was to fill in some of those gaps. We wanted to address 3 research questions. The first was to find out whether, if we sampled quantitatively, whether we could get some correspondence between the presence of snails at a site and the human infection burden at that site. And if we can, that means that a long-standing assumption is borne out, that it's primarily local exposure that puts people at risk, and therefore local control should be effective. Otherwise, maybe people are picking it up at the irrigation ditch down the street and then on their visit to their aunt's house last year, and that would change our approach to snail control. Second question, we wanted to test another long-standing assumption, that snails cluster in space. If they cluster in space, then Yahtzee for us, that's great. That would make them much easier to target, especially if those snail aggregations are persistent through time. And then finally, we had a hunch that it would be hard to sample snails in Senegal. Senegal's hot, our technicians can't touch the Schistosomiasis infested water all around them, and these snails are tiny and hard to find. But if snails are correlated with some other easy to measure environmental proxy, then maybe we can just target that particular part of the environment for snail control. So to summarize this part visually, we're saying snail density may not be correlated with human schistosomiasis cases if the snails are ephemeral and patchy and therefore difficult to quantify without sampling really intensively. If we do that intensive sampling, do we find this positive correlation with human schisto cases? And if not, maybe the presence of suitable snail habitat, maybe that habitat is just stable enough in space and time and an effective predictor of the presence of snails so that it provides a better indicator of human schistosomiasis cases than direct counts of snails might. Basically, we're asking what target gives us the most bang per buck, snails or their habitat. We tested each of these associations with a separate statistical test, and I'll walk you through each of those tests. First, we're going to look for correlations between human infections and snail density. Then we'll explore whether the variability in this relationship could be due to patterns of snail aggregation. We'll look to see whether there are any aspects of habitat that predict where snails are. And then we'll compete all these variables against one another and see who does better, snails, habitat, or some mixture of the two. Let's start at the very beginning. We set out, we worked in northwestern Senegal, a West African country with a very high burden of human schistosomiasis. We selected the region with the largest recorded outbreak of schisto in human history. The outbreak was set off by the construction of the Diama Dam here in 1986. The closing of the dam prevented saltwater intrusion into the upriver reaches of the Senegal and Lamsar rivers, which was really great for agriculture. And there's a lot of rice and sugar ag in that region now, but it also created perfect habitat for the schistosome competent snails. They increased in abundance, and there was a massive uptick in the prevalence of human schisto cases. We selected 32 sites distributed across 16 villages. So some of our villages have more than one place where the people go to access water. Our first task was to design a sampling strategy to quantitatively sample these guys. This is a tiny snail. We've got two species in our study region, Bulinus truncatus and Bulinus globosus. They're both intermediate host snails for Schistosoma hematobium, or urogenital schistosomiasis. And I'm just going to call them snails from here on out. We wanted to do this sampling in a way that was entirely quantitative. And we knew from some preliminary poking around at these sites that snail density was going to differ a lot from one microhabitat type to the next. So we started by taking aerial drone images of each site. Using those images, we drew a boundary around each site and then around the microhabitats represented at each site. We recognized three kinds of habitat: open mud, shown as open water in these pictures, emergent vegetation, or vegetation that's growing up out of the water, and then floating or non-emergent vegetation. We dropped a bunch of random sampling points within each site. We sampled 15 points at each site, and those points were distributed across the three microhabitats in proportion to their abundance at the site. So if a site was 2/3 mud, 10 of our 15 points would be in mud. We wanted each of those points to represent an exhaustive count of all the snails at that point. So we developed a new snail sampling method. We made this aluminum 3-sided enclosure, and we would drop it at the randomly selected point. The box enclosed all the vegetation, mud, and garbage within that point, and that allowed us to count every single snail within that quadrat. You're watching these guys deploy the quadrat in floating vegetation, but it works just as well in emergent vegetation, where we just cut back all the dry bits and examine all the vegetation matter that's underneath the water line. And it also works well in open mud. We can even attest to the resilience of this quadrat to being trampled by a herd of zebu. So everything in each of those quadrats gets transferred into a laundry tub and then thoroughly examined for snails. And to cap it all off, we take 3 scoops of the sediment at the end of each quadrat and pass it through a sieve to make sure that we've gotten every last snail. And in this way, we get an exhaustive, absolute estimate of snail density at each randomly selected point. Every single snail that we find gets counted, put in a vial, and transported back to the lab, where we confirm the species identity of the snail and assess whether they're infected with schistosomiasis. This is what the worms look like as they're spilling out of their snail host. This is the teeny tiny organism that is responsible for the suffering of 206 million people. So we went through this entire sampling process at all of our sites 6 times over the course of 2 years for a total of 1,900 quadrats and about 12,000 snails. We wanted to integrate over multiple seasons and years, so we sampled in the dry hot season, the rainy hot season, and the dry cold season across 2 years from 2016 to 2018. Simultaneously, we were assessing the infection status of the people who lived in the villages adjacent to our sampling sites. We had kids enrolled at each of our sampling sites, and we took urine samples from those children to assess whether and how many eggs were in their urine. And we did that 3 times over the course of the study. And our public health collaborators also administered praziquantel to any infected child. That meant that our estimates of human infection at the end of the study represent reinfection rates, just the worms that humans acquired over the course of the study. And that allows us to rule out the historical effects of previous residences and previous travel. So we end up with data on the abundance of snails on one hand and the infection burden in people on the other. And the first thing that we wanted to know was whether this quantitative sampling would let us find correlations between local snail abundance and local infections in people. And that would suggest that infections happen locally. But there were a number of snail-related predictors that we thought might be important, including snail density, or the number of snails per unit area, snail abundance, which is our best guess at the total number of snails at the site. It's snail density in each microhabitat multiplied by the area of each microhabitat summed across the entire site. We also had infected snail density and infected snail abundance, as well as snail prevalence. And then, of course, we wanted to include all the demographic predictors that we knew would be important in soaking up some of the variation in who gets infected and who doesn't, like sex, the age of our subjects, the number of people living in each village, and whether the village was situated on the lake or the river. Now, you've probably noticed that a lot of these variables are likely to be really collinear with one another because one is used to calculate the other, right? So snail prevalence times snail density is infected snail density. And obviously those variables can't all be included in one model because they will be too correlated with one another. So what we did in order to avoid this collinearity was to run all the possible non-linear or non-collinear combinations of those models. And then we took all those models and compared them with BIC to find which were the best ones at explaining the variability in human infection. We did this for two different suites of models. One suite of model were logistic generalized linear mixed models, which were for assessing the likelihood of infection. Are you infected, yes or no? And then the other suite of models were negative binomial generalized linear mixed models, which looked at the egg burden of infected individuals. If you're infected, how many eggs do you shed? So I'm going to show you these two sets of results separately. First for prevalence, or the likelihood that a person is infected, and then for egg burden, or the number of eggs passed by an infected individual. I'm only going to show you results for those models that fall within 10 delta BIC of the top model, which is why there are 6 models for likelihood of infection and 2 models for egg burden. Each model is shown with a different shape, and each model is going to contain a different combination of the variables that you see displayed on the y-axis. An odds ratio of 1 means no effect of a variable. And with that, there are a couple of very influential variables that we can pick out. We'll start with the demographic predictors. We know now that kids on the lake are way likelier to be infected than kids on the river. Boys were likelier to be infected than girls. No difference between small and large villages, and no difference in who is going to be infected between young kids and older kids. And then there are a few different snail and environment variables that come out as important. First, I want to draw your attention to the different variables that are going to be displayed here on this y-axis. This is already a little bit of a giveaway about what variables get retained in these 6 top models. You'll see that some of the variables are snail habitat variables, meaning variables that are calculated using both snail data and environment data. For example, snail abundance is the density of snails in each microhabitat multiplied by the area of each microhabitat at that site, summed across the 3 microhabitats. So it's our best guess at the total number of snails at the site. Snail variables, on the other hand, are just information about snails, just their density across microhabitats or their prevalence within microhabitats. And with all that in mind, we see that there are a handful of snail habitat and snail variables that are retained in our top 6 models, meaning that we do find some positive correlations between the presence of snails and the likelihood of human infection. If we then turn to the negative binomial models for egg burden in infected individuals, we get a similar answer. People on the lake pass way more eggs than people on the river. Boys pass more eggs than girls. Not much effect of village population density. Young kids seem to pass more eggs than older kids. In the models for egg burden, no snail variables are retained, but we do have a few snail habitat variables. So snail abundance is again important, as is infected snail abundance. Okay, cool. So we're on the right track here. Sites with snails put people at higher risk of getting infected and having heavy infections. And that suggests that snail control implemented at the scale of the local site could reduce burdens in local people. Now let's figure out the best way to target those snails. We've now found this correlation between human schisto cases and the local presence of snails. Let's see how variable the presence of those snails might be. We wanted to know whether snails aggregate, whether those aggregations are persistent in time, and how big the aggregations are. If snails aggregate, that's a really good opportunity to destroy them. If the aggregations are persistent, all the better, we'll always know where to find them. And if those aggregations are small, it'll take less time, money, and effort to wipe them out. For this analysis, we used the epidemiological software SATscan. And in SATscan, we selected retrospective space-time scans using discrete space-time permutation models, which is a big fancy way of saying that the model took a circular, varying diameter window and it ran it over the entire map of each one of our water access sites. And it judged that clusters occurred where there were more cases observed in the scanning window than the model would have expected under conditions of snails being randomly distributed across the site. Here's a summary of the output of our SATscan analysis. We've got each of our 6 time points on the x-axis, so you're moving through time as you move from left to right. All the sites are arrayed on the y-axis, and these are true to scale and grouped by village. The villages with the least site area are at the top, and the villages with the greatest site area are at the bottom. The pink circles represent snail clusters. Dark pink circles are significant, and light pink ones are non-significant. And the first thing that you'll notice is the total randomness of the distribution of these clusters. They pop up at one time point and they disappear at the next. There isn't a particular region of each site that's prone to clusters. We had 43 total clusters, with 30 of those being significant. Their average radius was 5.5 meters for a diameter of 11 meters. The average number of clusters per site over the 2 years of the study was 2. And here's some stats on their temporal persistence. 80% of our clusters were present at only a single time point. 13% of them persisted for 2 time points, and 7% of them were present at 3 time points, which was the longest duration that our model could detect. Yowzers, that analysis suggests that it's not going to be possible for us to go out and target aggregations of snails because they would be gone almost as soon as we measured them. And you have to constantly go out and resurvey to see where the next cluster has popped up. And as we learned, and as we'll discuss shortly, snail sampling is not a small feat. It's a big, big effort. This analysis suggests that you would need to do it over and over if you wanted to go out into the environment and target snail clusters. So we were disappointed, but we hoped that there might be some stable environmental correlate of snail presence that we could use instead of counting snails. So we found that these patterns of snail aggregation make the snail populations crazy variable. So let's now go in search of a proxy in the environment that's a little bit more stable and might indicate the presence of snails. So we wanted to identify environmental features that would predict the number of snails that we would find in a quadrat. And we had a bunch of predictors that we were interested in. There were 23 total environmental correlates included in these models. They included things like water quality, the microhabitat type, the specific species of vegetation found in each quadrat, and the amount of floating vegetation in grams found in each quadrat. And to assess this, we used delta generalized linear mixed models with forward and backward model selection based on BIC. And we ran these big fancy models, and a couple of parameters popped out as super important. What we found was that the presence of a couple species of non-emergent vegetation strongly predicted where the snails were going to be. Here you've got 3 species of non-emergent vegetation: Ceratophyllum, Ludwigia, and Potamogeton. And on the y-axis, you'll find the partial response of snail counts, so the number of snails adjusted for all the other junk in the final model. And it's very, very clear that these snails are associated with these particular types of non-emergent vegetation. Okay, cool. There are environmental predictors of snails. Now we just need to figure out if these are appropriate proxies for predicting where we're going to find human infections. So finally, we get to the big boss at the end of the game. What does a better job of predicting where human schisto cases are going to be? Is it the environment, or is it the presence of snails? For these models, we wanted to include both environmental proxies and the snail-related variables to see which were more powerful for predicting human infections. So we included all those snail-related variables that we talked about in the very first model iteration, but we also included site size, or the total boundary area of the site, the percent of each site that was covered in each microhabitat, the area of each microhabitat, and then of course those demographic predictors that we know are important for soaking up variation in who's infected and who isn't. Same collinearity problem, right? A lot of these variables are going to be correlated with one another because they are used to calculate one another. So we ran all the possible non-collinear combinations of these models and compared them with BIC. Again, these were split into two suites of models: logistic GLMMs for likelihood of infection and negative binomial GLMMs for egg burden. Okay, so we'll set this up exactly the same way that we did when we looked at whether just the Snail variables could predict human schistocases. We've got our results for likelihood of infection on the left and for the number of eggs these kids are passing on the right. And again, you'll see the variables retained in these models are displayed on the y-axis. And again, we're going to look at all the models that were within 10 delta BIC of the top model. You'll notice that I've divided up the variables: exclusively habitat variables with no snail information included. Then we've got snail habitat variables that contain information on both, and then just the snail variables. So let's see what we come up with here. Demographic predictors first. Just like before, kids on the lake are more likely to be infected than kids on the river. Boys likelier to be infected than girls. Small villages have about the same infection prevalence as large, and young kids are about as likely to be infected as older kids. And now we turn to the snail and environment variables. Notice that there are no snail variables retained in the models for likelihood of infection. Here we see only habitat or snail habitat variables. The important positive predictors of human schistocases are the total area of a site, the area of non-emergent vegetation at a site, the percent cover of non-emergent vegetation, and the mass of non-emergent vegetation. Snail abundance, which integrates information about the availability of non-emergent vegetation at a site, is also important. If we then turn to the negative binomial models for egg burden, we get a similar answer. Kids on the lake pass more eggs than kids on the river. Boys pass more eggs than girls. We have a small effect for smaller villages to pass more eggs than larger villages, and young kids pass more eggs than older kids. Again, we see that site area, area of emergent vegetation, percent cover of emergent vegetation, and mass of non-emergent vegetation are important. Snail prevalence, a snail variable, also ranks in the top models. So here's the upshot. Models containing habitat variables were better at predicting human schistocases than were models containing only snail variables. And if you'll indulge me a little bit, I just want to drive that point home by visualizing these models slightly differently. In this table, I'm showing you the results just for the likelihood of infection models, the models that predict whether or not an individual is infected. Each row is a different model. In the first compartment, you'll get all of the fit and the prediction metrics that we calculated for each model. For now, I just want you to pay attention to BIC because that's what we use to rank the models. You'll notice that all the models within 10 delta BIC of the top model are in the top compartment and the rest are in the bottom. The colored compartments indicate what variables were included in each model. All models had the demographic parameters, which are in gray. Some had habitat variables and snail habitat variables in green, and some had snail variables in orange. And what I want you to notice is that there were no snail variables included among the top models, only habitat and snail habitat variables. We can look at the same table for the egg burden models, the models that predict how many eggs an infected person is going to shed. And again, you see that snail variables were uncommon among those top models, and that habitat and snail habitat variables were uncommon among the bottom models. So our spatially intensive quantitative snail surveys did show positive correlations between where the snails are and where humans are infected. But there are several easier to quantify habitat proxies that actually did better at predicting where human infections are going to show up. And this is probably because snail populations are spatially and temporally variable, and any single measurement of their density is just a snapshot in time, whereas actual human burdens integrate risk over both space and time. The area available for colonization by the vegetation used by snails as habitat and the area actually covered by that vegetation at the time of sampling, those variables integrate that risk. And so they provide a more reliable indicator of human infection burdens. Okay, let's summarize. What did we learn here? Let's go through each of our research questions. Can quantitative sampling yield better correspondence between snail presence and human infection burden? Yeah, we found correlations between where the snails were and where human infections were. Do snails cluster in space? Yeah, they absolutely do. If they do, are those clusters sufficiently persistent in time that we can target them? Almost certainly not, which is a mega bummer. We wanted to be able to say yes here because we wanted to be able to go out and target these snails and destroy them. But the good news is that there are these environmental correlates, and we can make predictions about where human infections are going to be without doing all that work of counting snails. Even though habitat is a step removed from human infections, it actually does a better job at predicting where those human infections are likely to be. And that's a really lucky break because snail sampling is very labor intensive. This was an effort that involved thousands of person hours in the field, tens of thousands of dollars in salary and travel costs, and lots of specialized equipment. And we did some power analyses after the fact, and we figured out that we probably could not have gotten away with any less effort. And yet, we got a better answer from some of the easiest proxies to measure: site size and the presence of this non-emergent vegetation. And that's something that you can literally see from space. Monitoring these sites could be as simple, cheap, and fast as logging into Google Earth. Here you see one of our sites, and you can clearly make out emergent vegetation and floating vegetation under the water's surface. By measuring site size and the cover by non-emergent vegetation, anyone could develop a rough estimation of risk from the comfort of their own desk. And this is exactly what we need because schisto is a rural disease. Teams need to know where to go, both to distribute pills and to do snail control out in the environment. This prioritization of sites is a huge gap in the current control strategy in Senegal and throughout Africa. And if we wanted just slightly better spatial resolution, or if we wanted to control the timing and frequency of imagery and not depend on Google's satellites, a cheap drone does the trick. By doing these drone flyovers, we're able to calculate site size and percent cover of non-emergent vegetation to a really high degree of resolution. And hiring one guy to fly a drone over these sites is a lot faster and less labor intensive than hiring 10 guys to count snails. Our analysis suggests that it's also likelier to give you a reliable proxy of human infection burdens at that site. So two things drew me to research on infectious diseases. On one hand, the staggering diversity of parasites and pathogens, and on the other, the potential for us to harness knowledge of that diversity to alleviate human suffering. Schistosoma is a worm that shouldn't exist, right? It is improbably adapted to a lifestyle with long odds of survival, but it's also a parasite that's causing untold suffering among the world's most vulnerable people. And using ecology, we're developing ways to outmaneuver it. 206 million people have this disease and hundreds of millions live in constant risk of contracting it. We now have the ecological tools we need to protect these people from schistosomiasis, and for our team's part, this is just our first step. Thank you.
Thank you, Chelsea. Thank you very much. That's